Hello everyone, it is Dr. Miller Monfils, and we are going to do some very non-traditional teaching. I unfortunately have been in and out of the hospital, so I'm going to do kind of lecture videos like this for the rest of the semester. I'm planning on being in class, oh look, there's a pen growing out of my head, for this week and through the end, but one never knows with me. All right, so we are working on chapter 16, which is amino acids, and my cat is gonna jump up and say hello. So chapter 16, and I'll try to make sure that you guys can see all this. So just remember that DNA has RNA, and the RNA is what codes for amino acids. When you link amino acids together, that equals a protein. And proteins are just functional parts of the body, okay? And then special proteins are called enzymes. All enzymes end in the suffix of ase, and basically they are proteins that have a job, okay? So when we are looking at all of these things, again, you're gonna have to have like your textbook open and everything. And just also remember, some of the things are going to be givens. We're also gonna need to know the protein table, the amino acid tables, and things like that. So let's try to work through our chapter 16 questions, and hopefully you can see these okay. So there are 20 amino acids that are used to build proteins in the body. So it's asking you which are considered to have a non-polar R group. So remember that our R groups are just the variable ones. So that just means that it changes between amino acids, okay? And so remember we have that carboxyl group, right? And then we have a whole bunch of amino acids and stuff like that, right? But actually that was more for fatty acids. Take that away. Sorry, my brain is just a little bit scrambled too. So we're gonna have our backbone, which is going to be the same. And then this is going to be that R group. Okay, so the backbones are gonna have a polar head and a hydrophobic or a hydrophilic tail, right? Kind of the same thing. They link tail to butt, tail to butt. I like to kind of think of amino acids as ants, right? And so they all have a head and they all have a tail, and that's gonna be your backbone. And then this R group is what's gonna be changed. But why am I telling you this? Because then when they link together, it's gonna go tail, head, tail, head, tail, head, right? So just kind of think of that a little bit. And so when we're looking at that first question, it's saying which of the 20 amino acids used to, sorry, anxiety is now making me shake like a chihuahua. Which of the 20 amino acids to build proteins in the body, which of the following is considered to have a non-polar R group? And remember that our polar bears love the water. So they are the ones that end in sulfur, nitrogen, or oxygen. And again, your book kind of has some other things too, but those are the main ones. This is going to be found on page 551, okay? Our keyword that we're pulling out here is non-polar. So if we look at asparagine, I know that this ends in NH2. Cysteine ends in SH. Leucine is going to end in CH3, and glutamine ends in NH2. So again, that's on page 551 on my textbook, which unfortunately we found is a little bit off from yours. So the one is non-polar. Polar bears love the snow. Non-polar are the ones that are going to be scared of water, are hydrophobic ones. And so for this one, that's going to be leucine. Okay, so going on to the next question, it says the amino acid glutamic acid, which contains a carboxylic acid side chain, what is the overall charge on this molecule of a solution in the pH of 7? And so this is going to be talking about like the pKa, and that again is a value that you can look up. But remember that the pH of 7 is neutral, and then the carboxylic acid, that's gonna be the COO minus, and the pKa for COO COO minus is about 2, okay? So basically, if you have both a positive and a negative charge and they cancel each other out, this is obviously going to be 0. And so if both a positive and a negative are present, then that's called a Zwitter ion. Okay, so that's kind of just a fun thing too. But if we look at carboxylic acid, and again, I drew it out as COO minus, and so this -1 is going to be this B of -1. So I kind of told you a little bit of extra information that you didn't need to know, but it'll come into play later. All right. When the carboxyl group of glycine reacts with the amino group of valine, they form a peptide bond. What is the abbreviated name for the following dipeptide? So remember, di means 2, and then peptides are like the amino acids together. So basically it's asking you how you would react, or how you would shorten down glycine and valine. So the GLY, glycine, can be abbreviated GLY, and then the valine can be abbreviated VAL. So that would then give you glyval. So this would be like asparagine, serine, alanine, phenylene, valine, and glycine. So this one's backworks, but it's telling you again, remember how I just said it links head to tail, head to tail, head to tail. So this is what's going to be telling you why it's going to be glycine and valine, not valine and glycine. Okay. All right. For 16.4, so again, we just kind of have to pull out some of our keywords. It's the carboxyl group of alanine reacts with the amino group of cysteine, and the carboxyl group of cysteine reacts with the amino acid group, or the amino group of phenylalanine, and they form a tripeptide. So tri means 3, and peptides are linked, like 3 peptides are linked together. So it's asking you what amino acid is at the N terminal and the C terminal. And so the C terminal is going to be the free COO minus, or the COOH group, where the amino, or the N terminal, is going to be where you have the free NH3 positive. All right, and so then by looking all of these, and again, we have alanine, and we have cysteine, and we have phenylalanine. So by doing headbutt, headbutt, headbutt all together, you can tell that answer D is going to be the answer, because it's telling you which of the two are together, how you line them up, okay? All right, so moving on to the next question. What is the abbreviation for the following tripeptides? So we know that there's going to be 3 in there, and then it's histidinylphenylalanine. Okay, so histidine is 1, lucil is 1, and phe is 1. Okay, so it's going to be his, lu, phe. So that's going to be giving you C. And so again, you'll have, the amino acid chart, like there's not a gazillion of them. And so you can just be like, okay, where do these stop and start? And then again, link them together, butt to tail, butt to tail, butt to tail. All right, 16.6. What is the overall charge on a polar neutral amino acid when the pH is less than the pi? Okay. So this is kind of like a given thing. So with the pH, if the pH is less than the pi value, then that's going to give you a +1. Where if the pH is greater than the pi value, then that's going to give you the -1. And again, what I was saying before is that Zwitter ion has both positives and negatives charges. And so also in the backbone, there positives and negatives, and so there's an overall neutral charge. And so basically pH values above or below the point, the module will have a net charge. Okay, but again, it's asking you for polar neutral. amino acid when the pH is less than the PI. So again, pH is less than the PI. That's going to end up giving you a +1 value, which means that A is going to be your answer. Okay. For 16.7, the PI values of the amino acids, aspartic acid, glutamic acid, are around the pH of what? While the PI values of basic amino acids are typically what? Less than the pH. And so this is on slide 22 and slide 24. And so basically the answer for this one is going to be 3, and you can look those up again on those 2 slides. So the PI values of the acidic amino acids are around a pH of 3, where the PI values of those basic amino acids are typically higher than the physiologic pH. And again, on those slides that I sent out to you guys in chapter 16, it's on slide 22 and slide 24. All right, enzymes work with their R groups, and remember, R is going to be variable groups. So the backbone's all the same, and it's just that R group or that group to the side that's gonna be different, and that's gonna be the identifier of that actual amino acids. All right, so enzymes work when what? Now, I stressed this in the video, form is function, okay? So form is function. If they're not formed right, then they're not going to function right. Okay, so form is function. So this goes into when you're talking about, for instance, when you have a DNA mutation that can cause you to have like a polar amino acid change into like a non-polar amino acid or things like that. Because it's not going to fit in there the same, it's not going to fold the same. If it's not the right shape, it's not going to be able to fold together to actually have like the correct shape, and therefore it's not going to be able to function. So remember, form is function. So when you think of that and it says enzyme work together when their R groups can and fit into the active site of the substrate, yes, because that's how we know that, the actual enzymes, because remember, enzymes are proteins that have a job. And so enzymes are very much, it used to be called like the lock and key analogy. So it was only like one key for one lock. So if I have an enzyme here in a substrate, let's say this is a substrate and this is the enzyme, they're the same shape, so that would work because it's the same. But if I have the substrate here and my enzyme fits like this, even though it might fit, it's not actually making contact, so it's not going to be working well. So form is function, and it has to be the right shape in order to fit in there and function. This is also where inhibition comes in, and some of our medications and stuff that we use for pharmacologic intervention, because they will make something that has the same shape, so it'll fit into that active site, and therefore it will inhibit what is supposed to be getting in there, therefore blocking the potentially negative interaction, okay? So when we're looking at this, enzymes, again, form is function. They have to fit into the active site of the substrate, yes. They do form salt bridges with the substrate because that's how they're gonna connect and communicate with each other, so that is also true. And then they also do form hydrogen bonds with that substrate. So therefore, in this one, the answer is going to be D, in that all of them are correct. Okay, so moving on, enzymes are active at their most optimal pH. If the pH of the environment becomes too low or too acidic, which of the following is likely to occur? So remember, enzymes, they're proteins that have a job, and so they have to have good working conditions in order to function correctly. All right, so think of it. We used to do this experiment with crickets, right? And no crickets were harmed. But if you put a cricket in like the normal environment, they would chirp and they'd be fine. But then if you put it in like a freezer, they would almost go comatose, right? And they would stop chirping. It would still be crickets. And the activity of them would significantly decrease. Where if you put them in a warmer environment, their activity would increase. Okay, so just kind of think about that. They're little divas and they like to be in their most appropriate environment. Okay, so they're active at their optimum pH. This is also why fevers are bad for us, because if it gets too high, it can take out all of those active enzymes in our body, and therefore that would be why one would die with an extremely high fever or hypothermia, amongst other things too. So when if it becomes too high or too low, what's going to happen? Well, basically, is the enzyme going to become more active? Well, no, because it might be for short term, but honestly, it's not in that optimum situation, the environment. So no, that is not going to be correct. The R groups interactions are disrupted and no more catalysis occurs. That is correct, because we're going to be getting too hot. It's going to unravel. Mostly like heat, what it does is it denatures proteins. So there's a couple different things to think of here too. Denaturization or denaturing is just unfolding that protein, and usually that's at that tertiary structure. And we'll talk about the structures again right in the next question and stuff. But, if you unfold, then you're not formed and shaped correctly. Therefore, you cannot function correctly. Okay. So those interactions, they're going to unravel. They're no longer going to be formed correctly. So they're no longer going to be doing their job. The answer C is incorrect. The R group becomes supercharged, accelerating the reaction. No, basically, if you heat something up too high, you're gonna kill it, right? And then enzymes react by folding. Well, no, because they're already shaped correctly during the assembly of them, and they don't like get turned on or turned off by the folding, right? It's whether or not they're in the right environment, have the right substrate, and all of that. Okay. So the next question is going to be, the alpha helix represents which level of protein structure? So remember, our primary is, remember all of your P's. So your primary structure is like the pearl necklace, okay? It has peptides in with peptide bonds. And hopefully you can see what I'm writing over here, okay? And it's like a string of pearl necklaces. So they're all just kind of like in a strand. Secondary is like you give the pearl necklace to little Johnny and he starts to play with it, okay? And he's gonna wrap it around his finger, or he's gonna make snakes on a table. Those are alpha helices and beta pleated sheets. And so that's gonna be your primary and secondary, where you have alpha helices and beta pleated sheets. And so remember, primary and secondary interactions, these are all still backbone interactions. And that means that none of the R groups are interacting. It's just the backbone. And that's kind of what is causing like our alpha helix shape. Like when you look at a DNA double helix and it looks like a spiral staircase, it's all just these primary and secondary interactions, okay, between the backbone. Tertiary is when you say, okay, you have this pearl necklace, you take it away from little Johnny and you put it in your hand to kind of keep him from breaking it. And that's when all those hydrophobic carbons go hiding on the inside of it because they're scared of water. So the hydrophobic, scared of water carbon are going to be on the inside, where the hydrophilic the polar bears love water, they're going to be on the outside. Okay, so this is just a single necklace, so right? So it's a single sub-unit of SUB unit, and this is where it's going to be the R group interactions, okay? And then quaternary structure, that's just the fourth level. That's like if you take this wadded up pearl necklace that's in your hand and you put it in a jewelry box with multiple others, just to, have them just be stored there or hang out, right? So this is multiple subunits. And so this is when, again, the R groups are between the tertiary and quaternary. All right, so going back and we're looking at the question, it says the alpha helix represents what level of protein structure? Well, alpha helix was that keyword in the secondary structure. So that one is going to be answer B, which is secondary. Okay. All right, denaturization. This is a word that you need to know. Denaturing versus degradation. To denature something means just to unfold, kind of like I just said. But to degrade it means to break it down to absolutely everything. You can denature it down to the primary level, but so that means you're unfolding the quaternary, tertiary, and secondary level, but you're still going to have that peptide bond, that polypeptide strand in that primary strand, all of your P's, right? So that's denaturing. But we've all worked with a coworker that tries to degrade you down to nothing and rip you apart. So the only time that you would get rid of primary structure would be if you're degrading it, okay? So if we're looking at question 1611, right? Denaturing of a protein occurs when there's a change that disrupts what? Well, I said denaturing is everything but the primary. So it would be secondary, tertiary, and quaternary. So this one would be answer D. All right, for this one. Hopefully you can see it. I'm trying to do this upside down. All right, the tertiary structure of a protein is formed by all of the below interactions except what? Well, remember, this is when those side arms, those R groups are interacting, and we have those hydrophobic carbons, we have hydrogen bonds, and we also have hydrophilic interactions. So A, C, and D are all correct. So that means that the amide bonds are going to be the ones that are incorrect, okay? Because that's more at a different level, but that is definitely not going to be tertiary, because tertiary is that ball or that wadded pearl necklace in your hands, okay? All right, globular proteins have compact spherical shapes due to various interactions between those R groups or those side arms. Globular proteins are responsible for which of the following functions, all right? And this one is going to be, just kind of, you have to know what globular proteins are, but, it's the cell functions such as storage, transport, and metabolism. So I think of globe cells or globular cells. They're kind of like when you have, I always picture this little green alien thing that has like a globe, like for a goblet of wine to carry like mucus and things like that. I know that's very, very strange, but that's how I remember it. So globular proteins, they are going to be involved in cellular functions such as storage, transport, and metabolism. Cleaving an amide bond, that's going to have it's more than likely going to be an enzyme that's going to end in an ACE, right? And hydrophobic interactions, that's just carbons with each other that they hate water, so they hang out with each other, okay? All right, 16.4, in the dynamic model of enzyme reaction, the induced fit model, which is the one that we teach in this class. All right, this active site does what? And I kind of talked about it at the very beginning, right? Where it has to be the certain shape to kind of fit in there. So this one's called the lock and key mechanism, right? So it has to be the right key to fit in the right lock, all right? Or else it's not gonna work. So if it's not the right shape, it's not going to make that enzyme or that receptor function, right? And so the dynamic model of enzyme reaction, induced fit model, the active site is going to give it basically a little snuggle, and it's flexible, and it's going to adapt to the shape of the substrate. So while it's lock and key, yes, it fits in there, but then it actually has to wrap around it to actually make the reaction. So that's like the third step. First we have enzyme and substrate, then we have the enzyme substrate complex, then we have the enzyme product complex, and then we have the enzyme and the product is going to be step 4. But after the active site and the enzyme get together, then it wraps around it again just to really tighten that interaction and cause the enzymatic reaction to occur. All right, so that would be chapter 16.